Hi everyone and welcome to today's lecture. First we'll go over the chapter talking about the electron transport chain and oxidative phosphorylation, and then we will cover the chapter talking about photosynthesis. So first, starting with the electron transport chain. First I want to give a brief introduction of what exactly it is. So as you can see on this slide, the electron transport chain is basically a whole bunch of carriers that will pass electrons from NADH and FADH2 to oxygen. It's an exergonic process. And then oxygen, since we just said that they're passing the electrons to oxygen, oxygen is the final acceptor. Now, with this process, it's important to know the term oxidative phosphorylation because oxidative phosphorylation is basically where the bulk of ATP will be generated when we're talking about things like cellular respiration. And so oxidative phosphorylation will generate ATP by a pH gradient, basically the pumping of protons, which are hydrogen ions, and it will be pumping them across the inner mitochondrial membrane. So you can see that in this figure here. Across the inner mitochondrial membrane, you have the pumping of the pumping of hydrogen ions. And it basically ends up creating a pH gradient, which you can think of as a proton gradient, because there's a difference in the hydrogen ion concentrations in the mitochondrial matrix, which you see the arrow is pointing to this area, versus the intermembrane space, which is this gap here. And so that's why you see the protons moving across there, and that creation of a gradient and the pH gradient will basically allow for the generation of ATP that we're going to go through in later slides. Now, the mitochondrial structure is important for this process because, as you can see, we're focusing on two different areas within the mitochondrial structure. So by having this intermembrane space all around that portion of the mitochondria versus the central matrix, it allows for you to have that proton pump gradient because you'll be able to have high versus low concentrations of protons, and you'll be able to pump those protons across the gradient, okay? Now, when we say that you're having this passing of electrons from carriers, the 3 electron carriers that I want you to know, I listed out right here, are NADH, FADH2, and CoQ, which is coenzyme Q, which you may have seen that they sell as supplements that you can take. Now, when we are looking at electron carriers and electron transport, you may be wondering how we determine the direction, how that exact transport is kind of carried out. And so we can see here in these figures that they're able to map out the direction of electron transport by making these battery cells and looking at where electrons end up flowing. Okay, and you'll see terms like oxidizing potential, reducing potential, and you'll be able to map out which of the portions are being oxidized, what is getting reduced by looking at this. Okay, so when you create this battery cell, when you have electrons flowing away from something, toward, in this case, what they call the reference cell. So you have the sample side and the reference side. When you have the electrons flowing away from your sample, which in this case is ethanol, we use the little trick of Leo says Gur. So when you lose electrons, the Le, lose electrons, you're being oxidized. So ethanol, the electrons are flowing away from it. It's losing electrons. So the ethanol is getting oxidized, whereas over here on this side, the hydrogen side is getting reduced because it's gaining electrons, so it's reduced. And what you'll notice is because your sample is losing electrons and the electrons are flowing across the battery cell to the reference, you notice that the indicator here is showing negative 0.197 voltage. That's a negative because your sample is losing electrons, right? So it has a negative reducing potential. It has a low reducing potential because it's not the one getting reduced. It's getting oxidized, okay? So here on this side, now your sample is fumarate. We still have the same reference, which is hydrogen. And what do you notice? Now the electrons are flowing toward our sample. So fumarate now, you notice a positive on that reducing potential indicator, right? So when you see a positive here, okay, that shows you that your sample is getting reduced. It's gaining electrons. And so you can see the electrons are flowing toward the sample instead. Okay, so the trick is to remember that lose electrons oxidize, gain electrons GER, that's reduced. Okay, and then you can follow the electron flow to determine where there's a higher versus lower reducing potential. So now the electron transport chain is basically made up of 4 main complexes, and we're going to go one by one through each of these. So here in this figure, you see our first complex, complex number 1. So I want you to know that that one is called NADH CoQ oxidoreductase. Okay, be able to recognize that this term is the first complex of the electron transport chain. Kind of like its name suggests, this complex is responsible for transferring electrons from NADH through to CoQ, coenzyme Q. What's responsible for oxidizing in this reaction? So what's oxidizing NADH? Well, you see NADH, Leo says GER, right? So NADH is losing electrons here. They are going towards FMN. So FMN is oxidizing NADH, okay? Because it's the reason that NADH is losing its electrons. So it's serving as the intermediate to then carry those electrons to the CoQ over here, okay? So as you can see, this complex is an integral part of the intermembrane, so the inner mitochondrial membrane. It includes FMN, so that's the flavoprotein, which is flavin mononucleotide. So you could just think of it as FMN and that it's an example of a flavoprotein. You don't have to worry about the official name flavin mononucleotide. And that you have the transfer of electrons from NADH to CoQ as our first complex of the electron transport chain. Then we get to complex number 2, and complex number 2 is called succinate CoQ oxidoreductase. Okay, so make sure you recognize what each of the complexes are called. And this oxidoreductase complex is basically doing the same thing that the first one is, in that it's transferring electrons to CoQ, but instead of transferring from NADH the way the first one is, the second one is transferring electrons from FADH2 that have been produced by the oxidation of succinate to fumarate. Okay, now the substrate succinate is coming from the citric acid cycle, and then you get the conversion or the deoxidizing to fumarate by a flavin enzyme. Okay, and you also see FADH2 has been oxidized to FAD. And ultimately, CoQ is the one collecting these enzymes, these electrons. Then we get to the third complex. This one is called CoQH2 cytochrome C oxidoreductase. reductase, or sometimes you just see it as cytochrome reductase. This one's going to be catalyzing the oxidation of CoQ, as the electrons will be passed from CoQ along to cytochrome C in a multi-step process. And what I want to point out, what you can see in this figure, the difference between orange and green structures. Notice that each of the complexes that we've been talking about are shown in orange. They are completely embedded within the membrane, whereas the CoQ and the cytochrome C that are carrying or passing along the electrons, these electron carriers are not part of the complexes and can instead move around freely within or in the membrane. Okay, and so you're seeing we're still moving these electrons along. In the first complex, we were moving the electrons from NADH to CoQ. In the second one, the electrons were passing from FADH2 through that succinate oxidation to CoQ. Now we have CoQ passing those electrons that it gained. It's going to pass them to the cytochrome C, and then we can make it to the fourth complex. And now we make it to complex 4. This one is called cytochrome C oxidase. And it's easy to track what's happening across these complexes, especially with their names, if you keep remembering the Leo says Ger trick, right? So cytochrome C in the previous step was gaining electrons from CoQ, right? So that was cytochrome reductase, right? Gain electrons, reduced, Ger. So now cytochrome C is losing electrons because it's going to now pass them to the final electron acceptor, which is oxygen. So if it's losing electrons, Leo says Ger. So lose electrons, oxidize. So that's why this complex is called cytochrome C oxidase. Okay, so based on the name, you can kind of remember what's happening. If it's cytochrome C oxidase, cytochrome C is losing electrons. Who is it giving them to? The final acceptor of the electron transport chain, which is oxygen. And then we're right near the stage where we can have ATP synthase, then making some nice ATP for that cell. So now we're going to take a look at the energetics of what's going on. So everything we've been talking about so far is the electron transport chain. We did not get up to ATP synthesis yet with the oxidative phosphorylation or the ATP synthases. Right now we're still just focusing on the electron transport chain, which is the passing of those electrons from the initial NADH and the FADH2 stages of the complexes all the way down to our final acceptor of oxygen. So now when we look at this, when we look at the energetics of just the electron transport chain, you notice everything is highly negative, right? Near the end in terms of the overall reaction. So when you see negative, remember that means exergonic. So the flow of electrons through the electron transport chain from donors of NADH all the way down to the final acceptor, oxygen, that's an exergonic process. It releases energy, right? Whereas the synthesis of ATP in the next steps that we're going to talk about, that will be an endergonic process because remember, you always got to put in energy or you got to put in ATP to make ATP. Okay, but this table here is just showing the electron transport chain, so this is all heavily exergonic in terms of the overall energetics. Okay, so when we were finishing up the electron transport chain, I kept saying cytochrome C and cytochrome reductase and then cytochrome oxidase, and you're all like, what the heck is cytochrome? Where did that come from? So cytochrome is basically a conjugated protein. They contain heme groups, which, is the same structure that you'll find in hemoglobin and myoglobin, right? It's got the iron in the middle of the heme group. Now, the difference between cytochromes versus hemoglobin and myoglobin is that the iron of the heme and cytochromes is not for binding oxygen. Okay, instead, this iron is involved in redox reactions that we've already seen. Redox being reduction and oxidizing. Okay, so the iron of the heme in the cytochromes basically alternately gets, oxidized and reduced to then transport the electrons in the electron transport chain. And so there's multiple of these cytochromes, but I'm not going to make you memorize each of them. Just know, what cytochromes are, what their function is, the fact that they have the iron in the heme group that is for redox reactions instead of binding oxygen the way that it would be in hemoglobin or myoglobin. So now in the electron transport chain, we kept seeing electrons being passed around, but we didn't really get to the production of ATP yet. And we know that this is the point where you can make a whole lot of ATP for the cell. And that comes in with oxidative phosphorylation. Basically, thanks to this structure here, this is the ATP synthase, which is the protein complex that's going to be responsible for pumping out ATP for the cell. There are also terms that I want you to know known as uncouplers. Uncouplers are basically compounds that will inhibit the phosphorylation of the ADP without affecting electron transport, meaning that when uncouplers are present, ATP synthase will no longer be linked to electron transport. So when uncouplers are present, you can have those electrons transported and carried along those carriers, but you're not going to get ATP produced, okay? There are a lot of antibiotics that are uncouplers, like valinomycin or gramicidin A. And then basically when you have the the uncouplers present, you'll have oxygen still getting reduced to H2O. You'll have electrons moving along the electron transport chain, but you will not have ATP produced. And then once you remove the uncoupler, then ATP synthase will link back up to the electron transport system. There's another term that I want you to know, which is the PO ratio. That's basically to show the coupling of ATP production to electron transport. It's the ratio of ATP produced by oxidative phosphorylation to oxygen atoms consumed in electron transport. Okay, so the top of that ratio would be ATP produced by oxidative phosphorylation. The bottom of that ratio would be oxygen atoms consumed in electron transport. So now whenever you hear oxidative phosphorylation, you'll hear what's called chemiosmotic coupling. That's basically, chemiosmotic coupling and chemiosmosis is how you're really making all of that ATP with the processes that we're talking about here, because chemiosmosis coupling is coupling the protein gradient that we were mentioning before to ATP synthesis. Okay, it's the mechanism for coupling electron transport to oxidative phosphorylation, and it requires a proton gradient across the inner mitochondrial membrane that we saw diagrammed out in the earlier slides just a few minutes ago. And it is basically the main mechanism that we use to describe, how energy is made here. Now, what's interesting is that the way that the proton gradient will lead to the production of ATP will depend on ion channels through the mitochondrial membrane. And so the term that you see on this slide, ionophores, those are substances that will create channels for ions to pass through that mitochondrial membrane. And the uncouplers that we mentioned in the previous slide, antibiotics like gramicidin A and valinomycin, they are examples of ionophores. Okay, and so basically when we're looking at the idea of chemiosmosis, you can see the coupling to here, to the ATP synthase, where you're actually producing ATP. And so you have chemiosmosis, you have electron transport chain, you have oxidative phosphorylation. All of this is leading to ATP production. Now, when you see the ATP synthase, sometimes you'll see the terms F0 and F1. F0 just describes the portion of the synthase that is spanning across the membrane. And then F2, F1, so F0 is the portion spanning the membrane, and then F1 will be the portion that's actually using energy released from the gradient, the electrochemical proton gradient, to then phosphorylate the ADP to produce the ATP as the final goal. So when you hear F0 and F1, that's just the two portions of the ATP synthase, looking at, where exactly the ATP is being produced. So now this is another diagram of the ATP synthase. The F0 portion that I was mentioning before would be this blue portion that's embedded within the membrane. And then the F1 would be this portion sticking out of the membrane where you actually have the ATP produced. Now in this slide, we're showing that the chemiosmosis and the synthesis that we were talking about in the previous slide is not the only model, even though it is the main accepted model for ATP production. There's also the conformational coupling model. In this one, it's basically that the proton gradient is indirectly related to ATP production. And in this model, it basically says that ATP ends up released from this synthase structure as a result of conformational changes that have been caused by the proton gradient. So kind of like the conformation, the change in shape is causing it to spit out this ATP. And the way, textbooks like to describe it is similar to a turbine, and that's why it shows this arrow here. Basically, like this creates a rotor with the ATPase spinning around to harness a gradient of energy so that you can then have ATP produced. And so in that case, you would have basically, there's the chemical energy of the proton gradient, and there's the mechanical energy of the rotating the rotating ATP synthase protein. And so you would then have the mechanical energy converting to chemical energy in the form of the phosphate bonds of ATP, basically. Now, in addition to coupling, you'll also see the term shuttles, and these are carrier systems. They're very important because when we think about the processes that are occurring, there are a lot of different pathways that are all connected. Now, when we talk about glycolysis and you had that NADH produced, that was occurring in the cytosol. But then we said that the next steps as we go towards citric acid cycle, electron transport, all of that, we're then in the mitochondria. Now, unfortunately, NADH that's been produced in the cytosol by glycolysis cannot cross the mitochondrial membrane on its own. Okay? Instead, you need to have electrons transferred to a carrier to allow them to cross the membrane and make it to the electron transport chain. So these carriers are very important in the process in terms of the electron transport chain and ultimately ATP synthesis, right? Because you can't get to the ATP synthase without going through the electron transport chain first, okay? So ultimately, the number of ATP molecules generated during this process is dependent on the number of carriers or the types of carriers that are available, because that's going to be able to get the electrons from, the cytosol NADH to the mitochondrial membrane where you're going to have the electron transport chain. There are two examples that I want you to know for carrier systems. The first one is the glycerol phosphate shuttle, and the second one is the malate aspartate shuttle. The glycerol phosphate shuttle you'll find studied in insect flight mussels, whereas the malate aspartate shuttle you'll find within us because it's in mammalian kidneys, livers, and the heart. Okay, so these are just two examples. The first one, the glycerol phosphate shuttle, will use an FAD-dependent enzyme that's found on the mitochondrial membrane to oxidize glycerol phosphate. And it's basically a mechanism for transferring electrons from NADH in the cytosol to FADH2 in the mitochondria. The malate aspartate shuttle is a little bit more complex, and it's basically using the fact that malate is able to cross membranes, whereas oxaloacetate cannot. And we come across that in some of our other lesson slides as well, that concept that malate can cross membranes, but oxaloacetate cannot. Okay, and so basically the malate aspartate shuttle is a mechanism to pass electrons from the cytosol's NADH to NADH that's already present in terms of the mitochondria. Okay, so there have been a lot of different steps that we've been talking about recently, starting with glycolysis through prep citric acid and then electron transport and all of that oxidative phosphorylation chemiosmosis stuff. And so I threw in this slide, which is the table from your textbook that shows a breakdown of the ATP yield from the complete oxidation of glucose. And so I want to make note of a few things. I want you to notice that when we look at the glycolysis section, you'll see that glycolysis requires taking in some ATP to then make ATP. So you'll notice that it uses up ATP before yielding or giving you ATP. And so that's why the net yield from glycolysis is 2 ATP. So you see it cost the cell 2 ATP, it made 2 ATP, those canceled out, and then you're left with a net yield of 2 ATP. This stage here, pyruvate converted to acetyl-CoA, we usually call this the prep phase, as in preparatory. So it's kind of like how you have to cut up all of your vegetables and meats and all before you make a meal. You can't just, eat it as it is. You don't get anything from that. You have to cook it first. And so the prep stage, there's no ATP produced. It's just breaking down the molecules a little bit better to go into the citric acid cycle. The citric acid cycle will give 2 ATP. So we got 2 ATP from glycolysis, none from the pyruvate to acetyl-CoA prep stage. We got 2 ATP from the citric acid cycle, and then the big one is oxidative phosphorylation, where you can get around 30 ATP. You notice different shuttles yielded different amounts. Sometimes they show 30, sometimes 32, sometimes even some textbooks will show 34. Okay, so various amounts, but you can see oxidative phosphorylation is the stage where you get a whole lot of ATP produced. So now that is it for cellular respiration. We're going to move away from that, and we're going to focus now for the rest of the chapter or the lesson on the chapter of photosynthesis. Now, photosynthesis, we have the formula written out right here. So you can basically think of, well, what do plants do versus what do we do? So plants will take in the carbon dioxide that we breathe out. You got to water your plant. They got to take in some light, and boom, from that, they're able to make their sugar, their food, and at the same time, they let out some oxygen, which we then use. And I love this little meme, this cartoon that really shows, you're taking in the light, the carbon dioxide, some water, and he's letting out the oxygen and the glucose. Now, this will occur in the chloroplast, so circle star highlight that photosynthesis is taking place in the chloroplast, which is the organelle that our cells do not have, but plant cells will have. And there are 2 reaction stages involved that we're going to go into more detail with in the upcoming slides. There's the light reaction, and there is the Calvin cycle, also known as the dark reactions. Okay, and so with the light reactions, that's going to depend on solar energy being absorbed by chlorophyll to give the energy needed in those reactions. It's going to generate things like NADPH, which will then be needed in the next reaction, the dark reaction. Okay, and that dark reaction, or the Calvin cycle, that's where the fixation of carbon dioxide to actually yield the sugars is going to take place. And the reason why it's called the dark reactions is this one does not need to use solar energy directly. Okay, so instead it's using the products from the light reactions to get its things done to produce the sugar. Okay, so you see it's the fixation of carbon dioxide into sugar. Okay, so now even though I had introduced you to the light reactions and to the dark reactions on the previous slide, the dark reactions being the Calvin cycle reactions, I made this slide to highlight the key points of each to make it a little clearer for you. So as I mentioned on the previous slide, the light reactions are called light reactions because they're going to occur when solar energy is present, when the sunlight is available, whereas dark reactions, or what we call the Calvin cycle, they occur after the light reaction, and so they don't need the solar power to guide them. Instead, they're using the products of the light reaction. Now, the main purpose of the light reaction I highlight as the energy capturing reaction. So it's going to take that solar energy and it's going to convert it through passing electrons down the electron transport chain. It's going to convert the solar energy to chemical energy, and the chemical energy being ATP and NADPH that then go into the Calvin cycle. And in the Calvin cycle, that ATP and NADPH will guide or lead the conversion to carbohydrate, okay, to that sugar or glucose that is the final product in terms of all of that, all of the processes that we mapped out in the light and the Calvin cycles, okay? And in this Calvin cycle, the way that that carbohydrate is getting formed is through the fixation of carbon dioxide. Now, since I keep mentioning different reactions, different processes, and different locations in terms of the chloroplast within the previous couple of slides, I wanted to list out the parts of the chloroplast and the terminology that you'll see when we talk about photosynthesis. So as we already mentioned, the chloroplast is the organelle that you find in plants and not in our cells, and it will be the site of photosynthesis. Within the chloroplast, so we have the chloroplast here, you have a few different parts. You have the grana, which are the stacked bodies within the chloroplast containing the thylakoid discs. Okay, so each of those is the grana, and you'll see the individual thylakoid discs. These discs are very important because that's where the light reaction is going to take place, so the absorption of that solar energy. You will also notice that there's spaces in between each of the thylakoid discs. Okay, we call that the thylakoid space. And then you have the stroma, which you see highlighted here. And the stroma is where the Calvin cycle is going to take place. And that is basically the site of the actual production of the carbohydrate or the sugar in photosynthesis. So where you'll see the fixation of the carbon dioxide into the carbohydrate. So two that I really want to highlight here are the thylakoid discs and the stroma, because we highlighted on the previous slides the light reaction and the dark reactions, or the Calvin cycle. So please make sure to remember those two locations. Remember where the light reaction occurs and where the Calvin cycle or dark reaction occurs. Okay, so now that you're comfortable with the chloroplast, and we also keep mentioning chlorophyll, let's take a look at the chlorophyll structure. Now, this should look very, very similar to you. It looks very much like what we just saw a few slides ago when we looked at the cytochromes. Okay, and we had said that the cytochrome structure then looks like myoglobin and hemoglobin. So just like all of those guys, the chlorophyll has the heme structure, but now instead of having iron in it, or that Fe in the middle, the way that the other guys had, now instead you'll notice magnesium is in the middle. So circle star highlight that for chlorophyll, you'll have magnesium present in the heme group instead of having it be iron, okay? So it looks just like the heme groups of these guys, but now instead this structure has magnesium. And magnesium, when you go through various biology classes, you're going to see it's a very important cofactor. And when we talked about enzymes, we mentioned cofactors. Think factory, like the idea of, you know, those big smokes and metal and minerals and all, because cofactors are all the metals and minerals that help activate enzymes. So having magnesium present in the middle of this chlorophyll structure is very important, because of the fact that you're now going to need enzymes for photosynthesis. So the magnesium will help activate the necessary enzymes in this photosynthesis process. Okay. There's also another term that you encounter here called accessory pigments. They're basically going to play a role in photosynthesis, but they are not chlorophyll. So they will help with absorbing light and transferring energy to the chlorophylls that will then, you know, undergo the full photosynthetic process. Sometimes when you think of accessory pigments, you can kind of think of them as helping out chlorophyll. Okay. I also want to point out that when we're looking at the idea of chlorophyll, you'll notice in this figure there's a part called the reaction center. When light energy that comes in makes its way to the reaction center, that's when the actual chemical reactions of photosynthesis will begin. So this site is very important, this reaction site or this reaction center will be the site of basically a special pair of chlorophylls that are responsible for trapping light energy, the harnessing the solar energy. Okay, so I know some of this may sound a little repetitive, but I really want to solidify certain concepts in your mind. And we're going to dive a little bit deeper into some of the concepts that we've been mentioning so far. So the first area that we're going to go into again is the light reaction now. So we're going to zoom in on the light reaction that we keep mentioning. Once again, what's happening here, and I'm going to have it in a recap slide in a minute. The light reaction, the main idea is that you're going to have water converted to oxygen. So you see that over here. You see a photosystem 2 and photosystem 1. I know it's confusing that, the process is starting at photosystem 2 and moving to photosystem 1, but they're named based on when they were discovered. So they found photosystem 1 first. So in the light reaction, you see a couple things happening. You see water getting converted to oxygen by oxidation, and you at this point will also have NAD plus being reduced to NADPH, and you'll have basically redox reactions occurring, so combining reduction and oxidation. And in these reactions, you'll end up getting the coupling of the phosphorylation of ADP to ATP. And that is a process called photophosphorylation. So we're coupling the light reaction to the phosphorylation of ADP to produce ATP in photophosphorylation. Okay? And you're going to see that this whole light reaction is occurring in two main parts. So you have photosystem 2 and photosystem 1. So this is why you see the part that I mentioned before of water converting to oxygen is over here for photosystem 2. And then the part where I mentioned NADP plus being reduced to NADPH, you see that as a result of photosystem 1. Okay. And with these, I want to point out that the two photosystems are basically interacting with each other, and they're interacting indirectly because there is an electron transport chain between the two of them. And as you know, electron transport chains can couple with chemiosmosis for the synthesis of ATP. We saw that in the cellular respiration previously in the slides at the beginning of this lecture. And so you're seeing it again, an electron transport chain passing electrons to couple with chemiosmosis and produce ATP. And then what's important about, the results of this photosystem and the light reactions is that the products from here are then going to be able to go into the dark reactions. So the ATP that's produced and the NADPH that's produced by these light reactions will then lead into the dark reactions to give reducing power and energy for the fixation of carbon dioxide into the carbohydrate. Here is the recap of the notes that I was basically speaking on the previous slide. So now let's briefly zoom in on the individual parts of the light reaction. So first up, remember they're out of numerical order. So photosystem 2 actually comes before the events of photosystem 1. Photosystem 2 is where you have that oxidation of water that we saw in the previous slide to oxygen. So converting water to oxygen. This is accomplished by what's known as the oxygen evolving complex. And this is a very important step because this conversion of water to oxygen is the ultimate source of electrons in photosynthesis. And then those electrons are then passed through the electron transport chain that we saw between photosystem 2 and photosystem 1 in the figure on the previous slide. Now, when we are looking at the photosystem 2, that oxygen evolving complex that I just mentioned is basically a complex that's going to pass through a series of 5 oxidation states and will be transferring electrons in the process of, evolving that oxygen. And when we go through this photosystem, the first electron acceptor is a molecule called pheophytin. So you could just think of it as pheo. It's basically one of the accessory pigments that you'll find. And then what you have next is plastoquinone, which I have over here, this figure here. And you can think of that as PQ. It's very similar in structure to coenzyme Q or CoQ that we kept mentioning in previous slides for the other electron transport chain in respiration. And then this will ultimately lead to this guy here, plastocyanin, and that is linked to a copper protein. So it's again another structure that's very similar to the cytochromes that we had seen in previous slides, but now we have this idea of copper being with the protein instead of iron. Okay. Okay, so now that we've gotten past photosystem 2, we're going to focus on photosystem 1. You'll take a look over here. So we're focusing now on photosystem 1. The electron acceptors here are going to be chlorophyll A and ferredoxin, and basically the absorption of light by what's called P700, which is a type of chlorophyll, will then lead to a whole bunch of passing of electrons, and it's going to be, the light excites the P700 chlorophyll, gives an electron that's passed to chlorophyll A, and then you get the next electron acceptor being ferredoxin, which this guy here, that is an iron sulfur protein that's basically in the membrane of the photosystem 1. Now, what's important about photosystem 1 when we're looking at it, these electron transfers, the passing of the electrons, can be coupled to the production of ATP. And if you look in this figure here, you can see that this guy over here looks just like the ATP synthase that we saw after the electron transport chain in respiration when we were looking at the coupling of oxidative phosphorylation and also chemiosmosis. Okay, so it's valuable that you can have the coupling of photosystem 1's electron transport to the production of ATP. Okay, and you'll notice that when you have this, photosystem 2 is not involved at all. There's no oxygen produced here. This is only getting coupled to photosystem 1, which we saw was after the electrons are passed from photosystem 2. Okay. So now we're at the ATP production part of this process, and if you look at this and what's going on, like I said, even here in the previous slide when we were looking at this same structure, it's all very reminiscent of when we were looking at the electron transport chain in respiration and then the oxidative phosphorylation and chemiosmosis. So all of this is very reminiscent here now of the proton gradient and the proton pump in mitochondria. But in photosynthesis, what's leading to that proton gradient are the reactions that we've been going through so far. So for instance, in the beginning, the oxidation of water, then the electron transport chain, and finally that reduction of NADP plus to produce NADPH. And when you have that, when you have photosystem 1 reducing NADP plus by using hydrogen ions in the stroma to produce that NADPH, you end up with the pH of the thylakoid space becoming lower or more acidic than that of the stroma. So now you have that gradient of pH and of proton concentration, similar to what we saw when we were looking at the mitochondria. So now, as I've mentioned before in today's lecture, once you get to the dark reactions, which is that final portion of photosynthesis, also known as the Calvin cycle, what you end up happening here is carbon dioxide fixation. That's what's going on here. That's the main thing. And it results in the production of carbohydrates. And so a lot of times you'll see the carbohydrate shown as glucose, but that's not always the case. Sometimes it's sucrose or depending on which plant you're looking at. Sometimes it's polysaccharides like starch and cellulose, but usually they'll just show it as glucose. Now, when you have this carbon dioxide fixation and this whole cycle that you see starting off with the carbon dioxide entering it, the first enzyme is a very important one. You see that name come up. I just want you to know the name Rubisco is associated with being the first enzyme in the Calvin cycle. It's basically allowing for ribulose 1,5-bisphosphate to condense with carbon dioxide to form the 6-carbon intermediate. So they usually show it here as PGA or 3-PGA. And then you end up having the cycling going on. And then ultimately you end up here at Ruby P. And Ruby P or ribulose bisphosphate is the primary acceptor of CO2 in the Calvin cycle. Okay, and so ultimately you have it as a cycle because then you have the cycling of the materials. And basically they separate that into preparation, reshuffling, isomerization, and phosphorylation. But for our course, you don't have to know that. I just want you to be aware of some of the terminology associated with the Calvin cycle. So when you hear Rubisco, remember that is the first enzyme in the Calvin cycle, starting off the carbon dioxide fixation. You end up having the reduction taking place and regeneration, and you get Ruby P, which is the primary acceptor of carbon dioxide in this cycle. Okay, and so then the big thing over here, you'll notice the production of glucose, or like I said, depending on which plant it is, you may have different polysaccharides or disaccharides that end up getting made instead of glucose, but we'll show it here the way they do in the textbook as glucose. So now, even though the previous slide and the previous figure made the Calvin cycle sound very basic and straightforward and simple, the fact is, like most things in the sciences, especially in biochemistry, it is not, okay? So as you can see from this table, it's actually very complicated and involves about 15 different reactions just to end up at that one glucose or one carbohydrate at the end. So you need to know all of these reactions. Haha, just kidding. You don't have to memorize any of this. This is just to show you that it's a lot more complicated than we make it sound. So the last thing that I want to mention when it comes to photosynthesis is that there are alternative pathways. So in addition to the Calvin cycle, there's an alternative pathway for carbon dioxide fixation that happens in tropical plants. It's called the C4, so I wrote it over here, C4, and you see it down here, C4 pathway that you find in tropical plants, and it's named after the 4 carbon compounds that it involves. But basically in this pathway, carbon dioxide ends up reacting with what's called phosphoenol pyruvate to produce oxaloacetate, and oxaloacetate then is reduced to malate, a reaction that you've heard before in our lectures, and that gets transported from the mesophyll cells where it's produced and then ends up passed to the Calvin cycle. And basically, when you have C4 pathways, they've found that the plants end up growing more quickly and they produce more biomass. So if you think of tropical plants and you picture those huge, robust leaves and the ones that are just really big overgrowth type plants, it's because they're using the C4 pathway rather than being C3 plants, which will only be able to use the Calvin cycle. Okay, so C4 gets to use the C4 pathway and it then links up to the Calvin cycle, whereas C3 plants are just working on the Calvin cycle. Okay, whereas C4, you see how it connects to there, and this is that oxaloacetate and malate that conversion that I was talking about before. Okay, so make sure you're mainly aware of the name C4 pathway. That's an alternative to just using Calvin cycle that you find in C3 plants, and that it's found in tropical plants that end up growing much bigger and more robust, much quicker and more robust because of this pathway. And that is it for today. As always, if you have any questions, feel free to contact me day or night. You're never bothering me. Like I always say, I have no life, so never hesitate to ask any questions. Thank you and have a great day.